Hello, I am Shiro Jimi in Fukuoka University, Fukuoka, Japan. I am pleased to introduce the current result of our research. Today's talk is about beneficial effect of collagen dipeptide, such as proline hydroxyproline, Pro-Hyp, on wound healing using our established novel rodent model for scarring. I hope you enjoy my talk today. If you need to treat a new intractable disease in humans, the pathogenesis of the diseases should be clarified by using various scientific approaches, such as pathology, anatomy, physiology, bacteriology, and molecular biology, which may lead to new therapeutic solutions. In some diseases, pathogenesis is rather complex in vivo due to many physical factors. Animal models are thus important as preclinical study to clarify the pathogenesis under the effect of multi-factors that involve in vivo. Here you can see a cell that present among the collagen fibers. Our tissue cells are embedded in the collagen matrix. Even cells attach them during dynamic body movement and proceed with their task for tissue maintenance, such as migration, contraction, and proliferation. Collagens are thus not only an extracellular matrix protein, but also work as transmitter of tissue deformation to the cell. Then, cellular deformation may induce functional alteration by themselves. Today's talk is about collagen peptides. Therefore, it is highly possible that collagen breakdown products that are generated during wound healing may also play a regulatory role in wound healing. In this presentation, I will introduce the establishment of our scar animal models at the beginning and a possible prevention method for scarring at the end. In the wound healing process, when healing goes well after wounding, the damaged tissue would be healed and regenerated by the original tissue. In this case, it is fine. However, collagen accumulation is essential for the initial wound healing process, but sometimes could cause a problem such as scarring. And in more advanced cases, it becomes pathological scarring. Therefore, scar lesions are characterized by elevated acellular collagen-rich region in which pathological scarring may take place as a more advanced lesion. Pathological scarring with the excess collagen deposition may occur due to abundant tissue loss, intractable wounds, and wounds under higher tensile forces, or in some human races. The regions of higher tensile force are the anterior thoracic region, scapular region, and so on. Pathological scarring includes hypertrophic scar and keloids. The scar lesion often induce problems in medical, cosmetic, and QOL in patient. Keloid scars are more advanced and proliferative disease than hypertrophic scars. Then let's look at more detail about hypertrophic scars. The lesion may occur in everybody receiving traumatic injury. overproduction of collagen by activated myofibroblasts in the granulation tissue after wounding. The lesions are a result of prolonged infection and inflammation. The lesions occur under higher tensile forces. The patient samples of the skin lesions in our department are shown. Upper panel are HE stain and lower panel are MT stain, in which collagen are stained blue in color. They are normal skin and a general scar lesion. The scar lesion has more collagen accumulation in the dermis. In pathological scarring, hypertrophic scar and keloid. Fibrosis, more advanced and nodular region develop due to aggressive proliferation by mesenchymal cells. Study background. 1. Scarring is a form of healing that results in excess amount of collagen deposition by which normal healing and regeneration are inhibited. 2. In rodent, no animal models for scarring are available under physiological circumstances without any device and chemicals. 3. The relationship between scarring and regeneration is unclear. 4. Chemical or drugs to protect scarring are not available. The study aims. First, establishing a scar model in mice. Second, clarifying the relationship between scarring and regeneration using the model. Third, examining the therapeutic effect of collagen peptide Pro-Hyp on scarring and regeneration. First of all, I'm going to move the methodology of how to establish our scar model in mice. Now I'm going to introduce wound healing models in mice. If you make a skin wound without using devices and splints, the skin wounds in mice can quickly shrink and heal due to the movable loose skin nature. Therefore, we established the splinted wound model in 2017. In the mice, wound shrinking is prohibited by the splint, so this was used as humanized wound model. Also, no tension can be loaded on the granulation tissue by the splint. Thus, the model can be used as a tension-negative wound model in the present study. We had looked for the tissues that had tensile forces in the mouse body. The abdominal cavity is surrounded by muscle tissue without skeletal frame. Thus, we focused on abdominal muscle tissue that may always expose to muscle tension. We then measured tensile forces on the abdominal muscle wall without skin. When the abdominal wall was linearly resected in the horizontal or vertical direction, wound opened and tension for closing was measured. The tension in the longitudinal direction strongly loaded on the abdominal wall. It is because the muscle fibers run in the same direction from top to bottom. The method for creating the abdominal wall excision model, briefly, first, the abdominal skin was cut and opened. Second, the abdominal muscle wall was lifted up, and tiny part of the tissue was cut away. Third, the whole of muscle wall was covered by the skin to minimize external factors including air drying and inflammation from the skin. In this way, the wound on the muscle wall healed under the load of the tensile force of the abdominal muscle wall. During 21 days of the study, initially on day 0, a big hole present. Then the wound of the muscle wall became macroscopically smaller and finally invisible. Then developed granulation tissue are compared using the model with tension negative and tension positive state. In the back skin model with the tension, fibroblasts placed side by side in the granulation tissue from the beginning on day 7, and the structure are unchanged until the end of the study, whereas in the abdominal model, granulation tissue initially form a zonal structure similar to the back skin model, but become wavy and nodular with increase in collagen deposition after 14 days of the study. For the next step, we compare the extent of fibrosis with collagen deposition in the granulation tissue using empty stain. Fibrosis progressed over time in both models, but the level in the abdominal wall advanced extremely more than that in back skin model. Developed region in the abdominal model was unique, forming nodular structure and collagen matrices highly hyalinized. When developed granulation tissue were stained by picrocirrus red stain and observed under polarization microscope, collagen type 1 shown by yellow to orange color massively accumulated in the abdominal model, whereas type 3 collagen shown by green was distributed more in back skin model. The findings are consistent with collagen types in scar lesions and normal dermis tissue in humans. The result also suggests that matrix may be harder in the abdominal model than the back skin model. Such fibrous region remain as collagen-rich scar lesion even after 87 days of the study. The region may resist the regression process. In this study, we found many muscle regeneration in the granulation tissue, which was a big surprise for us. Therefore, the relationship between scarring and muscle regeneration in the abdominal wall was next examined. Muscle stem cells reside in the fascia. When the skeletal muscle is injured, mass regeneration may start, where the cell differentiated into myoblast, myotube, and myocyte, respectively. During the regression process, protein expressed time-dependently, including myogenin in the early phase and myoglobin in the late phase. Two representative lesions near the stamped muscle wall on day 21 are shown. You can see the yellow line. Yellow line indicates stamped original muscle wall. On the top of the original muscle wall, you can see cellularity is lower at the upper region and higher at the lower region. We then checked muscle cell regeneration in the granulation tissue by immunohistochemical detection using a myoglobin antibody. You can see the upper one is advanced fibrosis without muscle cell accumulation, and the lower one is advanced muscle regeneration. The relationship between fibrosis and muscle regeneration in the granulation tissue is significantly negatively correlated. We also check other elements in the granulation tissue near the stamped muscle wall. In the granulation area, CD31 positive endothelial cell of neovessels and alpha SMA positive myofibroblast increased, where myogenin positive muscle progenitor and myoglobin positive matured muscle cell invade. Electron microscopically, morphological characteristics of myotubes, myofibers, and muscle fibers are all detected in the newly formed granulation tissue. At the end of the presentation, I will show the result regarding the therapeutic challenge of using collagen dipeptide Pro-Hyp for scarring. The tissue picture from obese diabetic mice, namely DBDB mice. The granulation tissue developed on the back skin after skin excision. The mice have been used for model of wound healing delay under diabetic condition. We previously measured the collagen peptide content in the granulation tissue in normal and DBDB mice. Open columns are from distant dermal tissue in the same animals. Among collagen dipeptide, Pro-Hyp alone sharply increased over time in both mice. The increments well matched granulation tissue development. Moreover, DBDB mice show a significantly lower level than the normal mice. From these results, we speculated that collagen peptide Pro-Hyp might play a role in the development of the granulation tissue in wound healing. Then, we examined the effect of collagen dipeptide Pro-Hyp using our scar model in normal mice. Experimental procedure. First, the abdominal muscle wall was resected. Second, Pro-Hyp purity greater than 98% in saline was intraperitoneally injected at a dose of 500 nanomole per 200 microliter every day. Third, on days 7, 14, and 21, mice were sacrificed and used for pathological analysis. To know the transition efficiency of the IP injection of Pro-Hyp into the plasma, we measured the plasma level of Pro-Hyp after IP injection. After IP injection of Pro-Hyp, it appeared within 30 minutes in the plasma and then decreased by 3 hours. Thus, Pro-Hyp penetrate rather quickly over the vessel wall and get into the circulation, and may get to the wound lesions. The slide shows representative granulation tissues in the control and Pro-Hyp group on days 7, 14, and 21. Granulation tissue size in both groups changed similarly over time. The granulation tissue size once increased on day 14 and then decreased. However, fibrosis was suppressed in Pro-Hyp group. Significantly difference was found from day 14. The effect of Pro-Hyp were next compared. Fibrosis in the developed granulation tissue is shown in blue. Myogenin positive cells are in green, and myoglobin positive cells are red. Granulation tissue looks more active in the Pro-Hyp group than in the control group. Extent of myogenin and myoglobin expression density was then analyzed. Myogenic regeneration was accelerated by Pro-Hyp injection as compared to the control, especially at a late time. Summary of my talk today. We have established a novel mouse wound healing model that develops scar lesion at the abdominal muscle wall. In the abdominal muscle wall wound model, muscle regeneration started from the stamped muscle tissue, but it was prohibited by the development of fibrous scarring. Pro-Hyp administration after wounding decrease scarring and stimulate differentiation of the muscle stem cell for regeneration of the abdominal wall in wound healing. Conclusion. Pro-Hyp may work as a regenerative accelerator after wounding and is beneficial for scarless wound healing. Therefore, Pro-Hyp administration may help activate regenerative healing in the wound to be scarce. Finally, I will leave my message and comment for the future studies. We have to elucidate the mechanisms, how to commit Pro-Hyp in the scrap and build theory in wound healing. Explore the intracellular molecular mechanism of Pro-Hyp regarding mass regeneration and stem cell activation. Systematic and controlled human study should be performed. Thank you for your attention.